Bismillahirrahmanirrahim. Students, today we will discuss the male gametophyte in Ephedra. What is the structure and development of the male gametophyte? First of all, we see that in Ephedra, the microspores and megaspores, which are produced in the male cone and the female cone, these germinate to produce the male gametophyte and female gametophyte. Means that microspores, these produce the male gametophyte, and megaspores develop the female gametophyte. Male gametophyte is endosporic, means the microspores, they start their development before they are shed from the microsporangia. The microspores, they start their germination, they start to germinate before they are released from the microsporangia, means when they are inside the microsporangia, they start their germination. In last lecture, we discussed that in microsporangia, microspores are produced, and the structure of microspore is these are oval in shape, these are more elongated than their width, and on their outer wall the parallel ridges are present on their outer wall. Each microspore is surrounded by a two-layered wall. The outer wall is known as exine, which is thick layer, and inner to exine is intine, which is delicate and thin layer. And the nucleus of the microspore, it is shifted towards one pole of the microspore before germination. Now we see the male gametophyte development. Before pollination, the development of gametophyte is started when the nucleus of the microspore is shifted towards one pole of the microspore. Then it divides to form prothelial cell nucleus, which is surrounded by a wall to form a small cell, which is known as the first prothelial cell, present near one pole. Then the nucleus of the larger cell, it divides mitotically to form a small second prothelial nucleus and a larger nucleus, means two prothelial nuclei are produced. One is surrounded by the wall, prothelial cell, and the second is not surrounded by wall, and it is prothelial nucleus. Then the larger nucleus divides again to form two nuclei, number one generative nucleus and the number two tube nucleus. The generative nucleus divides to produce two nuclei again, number one the stalk nucleus and a body nucleus. All these nuclei, they are not surrounded by a definite wall. No proper wall is present, but these are separated by the cytoplasmic sheaths. Now this microspore is at 5 cell stage, 5 nucleate stage, 2 prothelial nuclei, 1 stalk cell, one body nucleus and one tube nucleus. Now this semi-germinated stage of the microspore is known as pollen grain, means this five-celled semi-germinated stage of the microspore is known as pollen grain, and this is the male gametophyte, the development of the male gametophyte before pollination. And this development to five-cell stage, it occurs when the microspores are inside the microsporangia. These are not released from the microsporangia up till this development. We can see here the diagrams for the germination of the microspore. This is the microspore. Nucleus has been shifted to one pole. This is first prothelial cell and large nucleus. Then the second prothelial nucleus is produced and the larger nucleus. This larger nucleus divides to produce the tube nucleus and generative nucleus or enteridial cell. And then next, this generative nucleus divides again to produce the body cell or spermatogenous cell and a stalk cell. And at this stage, the male gametophyte at this 5-cell stage, this is known as the pollen grain. And at this stage, after this it is released from the microsporangia. Here again we can see the diagrams of the development of male gametophyte before pollination. Microspore nucleus divides to produce first prothelial cell surrounded by the wall and larger nucleus. It divides again to form two nuclei. Smaller nucleus form the second prothelial nucleus, and the larger nucleus, which is known as the enteridial initial nucleus also, it divides to form the stalk cell and the to form the two nuclei, generative nucleus and the tube nucleus. Then this generative nucleus divides again to two nuclei. One is the stalk nucleus and the other is the body nucleus. And at this 5-cell stage, this male gametophyte is known as the pollen grain, and this development occurs in the microsporangium. After this, these are released from the microsporangia. Then the development of the male gametophyte after pollination, when this male gametophyte or pollen grain, it reaches to the female gametophyte, then within the female gametophyte inside the pollen chamber, the exine of the microspore, it is released, it is removed, and the pollen tube is produced. Pollen tube arises, and meanwhile the body cell nucleus, it divides to form two nuclei, which are now known as the male nuclei, male gamete nuclei, and these may be slightly unequal in size. The tube nucleus and the male nuclei, these are migrated into the pollen tube, means these enter the pollen tube of the male gametophyte. In the pollen grain, the prothelial cell and the stalk cell is present, and then the body nucleus in the male gametophyte, after pollination, it divides to produce the two male nuclei, and pollen tube has been produced before the production of the male nuclei. And then in this pollen tube, which is produced after the removal of the exine of the pollen grain, in this pollen tube nucleus and the male nuclei enters, and fertilization occurs, and then they reach to the female gamete for fertilization. Here is the graphic representation of the development of male gametophyte. Microspore nucleus divides to form the first prothelial nucleus, which is surrounded by a wall to form the prothelial cell, and a large nucleus. Then this large nucleus again divides to produce two nuclei, second prothelial nucleus, which is not surrounded by the wall, and a large nucleus. Then this large nucleus again divides to form two nuclei, tube nucleus and the generative nucleus. And then this generative nucleus again divides to form the two nuclei, stalk nucleus and the body nucleus. At this stage, the 5 nuclei are produced, and at this 5 nuclear stage, the microspore, now known as the pollen grain, and the male gametophyte is at this 5 cell stage, semi-germinated. This male gametophyte, it is then released from the microsporangia after this 5 cell stage, and this development, it occurs when the microspore it is inside the microsporangium. Then further development of the male gametophyte, it occurs when this pollen grain reaches to the female gametophyte, and in female gametophyte, when it reaches to the pollen chamber, then pollen tube is produced, and the body nucleus, it divides to form the two nuclei, these which are known as the male nuclei. And then in the pollen tube, the tube nucleus and this tube nucleus and these two male nuclei, these first entered into the pollen tube, and this is the development of the male gametophyte, which occurs after pollination. So today we have discussed the structure and development of the male gametophyte in Ephedra, and next we will discuss the female cones and the ovule structure, inshallah.